We pulled together a short group of sample prep experts that will do their best to answer your sample prep questions related to genome generation. Your hosts will be Michelle Kim from Circulomics and Christa Caneda from Arima Genomics. Welcome, ladies.
Hello.
Hi. Thanks for coming to the sample prep panel.
Thank you.
So you'll be able to learn from and ask a question to one of our illustrious experts. I'm Michelle Kim. I'm lead scientist at Circulomics, and I develop and think a lot about sample prep to help you guys get good DNA. And my co-moderator is Christa Caneda at Arima Genomics.
Hello, yes, my name is Christa Caneda. I am an associate scientist at Arima Genomics, and I am the lead project manager for the services department here. So lots of experience with lots of different biosamples here at Arima as well.
Great, and today our expert panelists are Maggie Weitzman, who is at the University of Oregon Genomics and Cell Characterization Core Facility, Tanya Murtha at the Iowa State DNA Facility, and Jennifer Balacco at the Rockefeller University Vertebrate Genome Lab. So welcome everybody. I'm so glad we could all be here together. And I thought we would, we'd love to hear from you guys. We have, Christa and I have a few questions we'd like to ask you so we can get a lead into your expertise, and then we'll open up the session to the Q&A from the audience. I guess one of the questions that I get a lot from our customers have to do with samples that are challenging. So what are the aspects of challenging samples that you find are the most difficult obstacles?
So
to any of you.
One thing that I find most challenging when I'm prepping a sample that I've never worked with before is looking at its genome size. The correlation of the genome size is kind of important when you're working with the Circulomics extraction kits. I find the smaller the genome, the more mass you have to start with to get a higher yield at the end.
Great, and Jennifer or Maggie?
Yeah, absolutely. So kind of on that idea, we deal with so many different wonderful types of tissue types. And so when you go in, it's hard to optimize if you haven't worked with that before. So the more that we know about a tissue type going in can really help us to best pick a protocol for isolating high molecular weight DNA at the proper yield.
Yeah, I agree with my panel mates. Knowing as much as possible about the sample can inform where you want to start in your protocol optimization, which can save you a lot of time. There are certain organisms or species that I found, I just have not yet found a protocol that works for them. So there are particular species where I just haven't figured it out yet. So there are those out there.
Oh, I love it. I'm kind of going off the conversation of challenging sample types. Tanya touched based on smaller genome sizes. Are there really, and you're going into different species, are there certain types of samples that you find particularly challenging other than the genome size limitation?
I find anything that is aquatic to be kind of challenging. It's like they have secondary metabolites that kind of make it more difficult to get a high yield or high molecular weight DNA. Recently, I extracted from some abalone species, and while they weren't super high molecular weight, they were maybe 40 to 50 kb, the yields were really, really small, but fortunately it was enough to run a couple flow cells on the GridION.
Yeah, I have struggled with a few sea creatures myself as well. I tend to find that fish and crustaceans are on the easy end of sea creatures, but things like jellyfish, I just haven't figured it out yet. It could be secondary metabolites that are inhibiting extraction or purification of the DNA, but I also think it's a factor of that these organisms are mostly made of water and they don't have a lot of DNA. Like the nucleus mass per animal mass is very low, because I found the same issues when trying to do chanterelle mushrooms, which are also mostly made of water, very low DNA content compared to like a liver or a kidney. And so for me, like the watery organisms are the hardest ones to extract just because they have so little there. And any sort of inhibitors, metabolites can chew up what's left and you end up with low quality, low yield DNA. So those ones I haven't figured out yet.
Yeah, similarly to that, fish skeletal muscle has always given us issues with yield. So generally we have to change the input to be up to 100 to 200 milligrams to have yield from extraction, as well as anything that's very fibrous. So if you're dealing with a skin tissue or other skeletal muscle in general, or something that's pigmented, can be really challenging.
Yes, I have found that to be the case as well. What do you guys like to use for your sample disruption methods?
My favorite is the QIAGEN Tissue Rupture 2. It's basically an immersion blender for tissues, and it just makes like a really frothy smoothie of whatever you've got. And for tougher tissues like skin, I use that as well, but I just make a really, really fine mince with a razor blade first and sort of put like a paste into it, and that seems to work really well. That's my favorite.
I agree with Maggie. We use the Tissue Rupture 2 quite often, but for certain tissue types, we modify, and we're trying out using a pellet pestle, thanks to Michelle Kim recommending this. So this also comes up with low input samples where we're afraid of losing some of the tissue or DNA that might end up getting stuck to the Tissue Rupture probe. With a pellet pestle, it can be a little bit easier to wash that and really preserve and prevent loss of your sample.
I agree with that. We don't have the tissue ruptors, so I do everything with a pellet pistol. That seems to be working pretty decently, although I do find that in the protocol it says to smush it like 20 times, 20x or 10 times. I go pretty above and beyond that, because if I can see it visually, it's like, oh, just got to keep going. So I find that I have to go above and beyond that step.
Oh, that's a great point. And especially when we work with insect tissues, when we have like insects with high exoskeleton content, those are very troublesome insects to get the DNA from. So we at Arima, we like to use the Tissue Rupture 2 from the Biomass 2 from diagnosine, in addition to the mortar and pestle, and we've had good experience with that for some lower yield samples.
Sorry, go ahead.
Yes, but in addition to, I guess, in addition to disrupting tissue samples, do you have any typical or favorite methods for just general extraction?
Circulomics is my favorite. We struggled a lot with DNA extraction. So I do all the pack biopreps in our core facility, and extraction was our pain point for years when we first started PacBio. We tried CTAB, we tried agarose gel plugs, we tried magnetic beads, everything you could think of. And then I don't know, a little bird told me to try Circulomics, and it really just changed everything. It's so easy, it's fast, high yield, high quality, high quantity. It's just some things, you still have to go back to CTAB, like plants. Although I did notice on Kelvin's screen that your plant kit says that tissue is now going to be used instead of nuclei isolation. So I'm really excited about that. And maybe Michelle, you can tell us more about it.
Yes, my colleague Renee Fedak is working on the total plant tissue extraction method, and I think we'll be releasing that soon. But yeah, I'm glad that our kits are working out well for you. Makes me very happy.
I agree 100% with Maggie. We implemented a DNA extraction service at the facility because people are really having a hard time getting, honestly, greater than 10 kb fragments, and which we all know that's not quite up to par anymore. So we almost exclusively use Circulomics extraction kits. Before I started with Circulomics, I was using some QIAGEN kits, but I have noticed that the Circulomics ones are so much easier to use. It's so much more reliable. And it's honestly part of my favorite part of my job is doing extractions with these kits. So yay, Circulomics.
Oh, I love that.
So for us, historically, we were using agarose plug extraction methods kind of as our gold standard for high molecular weight DNA, but we've also moved almost exclusively to the nanobind. It really saves us time, and we see great results.
Wonderful. Yay. I swear we have not paid any of our panelists to say these things.
We just really love it. It's changed everything for us.
That's great. I love to hear it. Actually, I got to say, I've been here for a lot of years now, and I still really like doing the extractions and seeing the nanobind attached to the DNA. All right, so I guess one thing that Christa and I have wondered is, do you see that different sample prep methods have different effects on different downstream applications?
I have seen that, like comparing some QIAGEN, their genomic tip extraction kit doesn't really see the downstream effects of lesser throughput for like GridION sequencing, but some of their kits I've noticed that there's one specifically with magnets, I can't recall the name, but it does decrease the throughput. And for Circulomics, which is why we're almost exclusively using Circulomics now, is we don't see that decrease in throughput when you have an increase in high molecular weight DNA.
I found that as long as the methods get you the same size distribution, that they don't really have much of a difference on like sequencing performance. So like an old school like DNA precipitation spooling on a glass rod can give you really, really high quality DNA, but it doesn't necessarily sequence any better in my experience. And so the ease of using a kit that's much simpler and can still give you high quality is worth it. So basically it's not worth doing the more complicated, really, really long, high extraction, high molecular weight DNA preps because it doesn't sequence any better in my opinion.
Yeah, we've seen that as well. So what we aim for is ultra high molecular weight DNA. So however you can get there is really the best way.
Oh, perfect.
All right, I get close to the camera now. I can,
So,
all right, I am going to open up the Q&A from the audience. Let's see, our first question is from anonymous attendee, actually. I have recurring DNA quality problems, 26280 problems, when using Ampere beads. Is there any data available when traditional beads-based purification versus nanobind?
I don't have any data, although I will say that I go to nanobind when I can't clean something up with beads. So the nanobind seems to be superior in improving the purity compared to beads. It's also a lot easier to handle that super high molecular weight DNA on a disc than on a bead. So that's really helpful as well.
And then if you're having quality issues, I also like to use the short read eliminator kit. I think that increases the quality. And for some species, the OD readings don't really they don't really seem to make a difference when it comes to sequencing. I've sequenced some with 260, 280 down to like 1.3, and it sequenced just fine. The quality scores were just fine. And sometimes it seems like those are almost irrelevant numbers sometimes.
I totally agree. I think that it's a lot less, maybe they've improved the chemistry of like PacBio sequencing in recent years, but it used to have more of an impact than it does these days. And so if I can't get something above like a 1.5, I just prep it anyway and it usually works fine.
I'd like to also add, I agree with my panelists, but some tissue types will always just give you these weird readings that aren't ideal, and downstream, it often doesn't matter, but it all depends on what technology you're planning on using.
Oh, wonderful insight. And kind of, and for the next question from Andrea Otessen, have you compared any sort of inter-kit comparisons between Circulomics and Zymo or Promega?
I have not personally tried those other kits. I've tried QIAGEN kits, the high molecular weight QIAGEN kit, and Circulomics wins every day.
I agree with that. I've used the Zymo kit, but that was for 16S, so that's really not comparable in this situation.
I see.
Okay, we have a question now from Yelena Petrovic. What antibodies does Arima high chip kit supports? It looks like it is only validated for histone modifications. Is that right?
Oh, that is a great question. So currently we are validated for histone acetylation and methylation. I know that the product development team is hard at work for a lot of the, they're doing a screening for a lot of antibodies that have been specifically requested and surveyed by our customers. So we're hoping to expand that catalog shortly. Oh, But thank you for the Arima question. To go bring me back to the panel, is there any tips for those doing sample prep in the low biomass world?
Yeah, so we've been experimenting with working with organisms that are quite small, including some single insect, single arthropod. And we see that one of the things that you can do is change your disruption method. So as mentioned earlier, instead of using something like a tissue ruptor where you can lose a lot of your input sample, you can try to use something like a pellet pestle, or try to minimize the number of times you're mixing with something like a pipette tip, or things like not changing your tubes as frequently. Any step along the way that you can see your protocol where you can prevent loss is ideal.
I have.
Oh, sorry.
Go ahead. No, please go ahead.
We have experimented a little bit with these little bags that they're made of plastic, and you can freeze them in liquid nitrogen. So you can put your sample in a bag and then attach like a glass vial to collect it. And you basically smash the tissue inside of that. So you lose a lot less material than you would on a mortar pestle. And so as you smash inside the bag, you can kind of just tip it upright and the powder will collect in your vial. So that can work. I try not to do things under liquid nitrogen because it's so much more work, but it can help when you have a low sample and you really don't want to lose any inside of a mortar and pestle or something like that. I think, are they called cryo prep tubes?
It is, yes.
Yes. Okay, you guys know what I'm talking about. We don't have the machine. We just smash it with a hammer. We freeze it in liquid nitrogen, and then we put it on like a cold metal block that's sitting in dry ice. So it's really cold, and then we smash it, and then we use the powder that way.
Yeah, it's a clever workaround.
Cheaper anyway.
Yeah. Yeah. All right, let's see. Our next question is from Vivian O'Donnell. Lately, I compared magnetic beads, MagMath PathKit versus Circulomic Nanobind, blood sample extraction for viruses. Nanobind gave excellent results, high yield. Oh wait, I'm sorry, this is not a question. It says excellent results, high yield, great quality, very much looking to keep working with them. Thanks.
I'm just excited.
All right, so that was your question. Next one, actually a question. Tiny insects, which we have touched already. Tough chitin exoskeleton, stabilized cross-linked DNA chromatin first, or crush mechanical pulverized specimen first. Chitin digestion enzymes.
I have not personally experimented with any of those techniques for cross-linking prior to extraction. Personally, I'd rather not. I worry about damage to the DNA with doing that. So I'd rather keep it a little more like it was in the biological system and not introduce chemicals that could inhibit sequencing. I've never even considered that. Has anybody else tried that?
That is a great question. We work with both different types of insects at Arima, whether that be mosquitoes, flies, or different types of beetles. Especially working with beetles, we end up, it's actually a little bit of a bit of both, actually. I think in the end, the answer is yes. Where first we will take a razor blade just to kind of go through the exoskeleton layer, just to expose the inner organs of the insect. From there, we'll cross-link, and then we'll do a more intensive pulverization that kind of furthers the more gentle cold razor blade method. So we do mechanical, then cross-link, then pulverize. So that's how we've been approaching it, but we haven't really looked into any sort of digestion enzymes for these insects.
Tanya or Jen, do you have anything to add to that?
I've extracted from mammalian chewing lice, so those are super tiny insects, and I didn't do any digestive anything first. I just mechanical pulverization, and you have to know when you're using little insects, and with the Circulomics protocol, they float, so it's really difficult to actually get them down to the bottom of the tube to kind of squish them up. But I followed the protocol similarly to the Drosophila protocol that Circulomics has. And from 30 milligrams of mammalian chewing lice, I got about a microgram of high molecular weight DNA. So that was just enough for one flow cell. And with a an organism with such a small genome, that was plenty for the researcher.
Oh, perfect.
Great.
Fantastic. And I guess we've been talking a lot about genomic DNA. Do you have any recommendations when it comes to RNA, especially RNA viruses?
Just RNA extraction in general for long read prep, like for iso-seq?
Yes,
or...
yes. Specific recommendations for an extraction method to use, namely with RNA viruses.
Well, we use a lot of Zymo kits for RNA extractions. I haven't tried their long read kit, but their direct cell kit works great. I can't vouch for it specifically for viruses or for long read applications, but definitely vouch for them for Illumina preps. So they do isolate very high quality intact RNA, which I think would be suitable for downstream long read applications like iso-seq on PacBio.
And for us, for our PacBio iso-seq, we usually use a QIAGEN RNA Easy kit, which works really well, but that's not for RNA viruses.
Unfortunately, we don't do any sort of long read RNA sequencing at Iowa State right now, so I don't have anything of value to add to this.
Okay. Our next question is from Chris Christensen. Are there any tips for working with fish or marine creatures?
I would say go for the squishy organs is my recommendation. So avoid the fin clips and the skin and the bone. If you can, go for liver, heart, kidney, something like that that's more easily pulverized. But I found that fish and crustaceans work pretty well with the Circulomics extraction kit. Not so much luck with the watery things like jellyfish. I'm actually about to start an octopus project, so we'll see how that goes. Hopefully better than the jellyfish.
Oh, fingers crossed.
Yeah.
I recently extracted DNA from a marine sea sponge, and I used some of the tougher portions, like Maggie was saying, those don't really work out as great, like the skeletal part, but the reproductive organs, those had produced much better results. So I would say maybe lean towards reproductive tracts for sea creatures if you can.
And for cartilaginous and bony fishes, we see great results from tissues like heart, as well as if you're less concerned with contamination, gills also are very high yield and can provide ultra high molecular weight DNA as well.
Tanya, do you mind if I ask what extraction method you used for the sponge you said?
Yeah, the sea sponge. I use Circulomics.
Okay.
Yep.
Great.
Yes.
It works for everything.
It does.
Oh, fantastic. And all right, so the next question from Sarah Frampton. She asks, for nanopore sequencing of DNA from blood, do you have any reason why the high salt extraction method isn't as popular as the phenylchloroform one, which would be a cheaper alternative?
If I had to guess, people are just moving away from the phenylchloroform because they're hazardous chemicals. You have to use the fume hood. I don't have any direct experience with either one for nanopore sequencing of blood, but if I had to choose between two, I would go with a non-hazardous version.
Oh yes, that comes to mind as well. Definitely just getting away from the hazards are always just easier for the facility and ourselves.
I think we might have skipped a question about blue pippins.
Yes, we did. I just realized that. My apologies.
Okay.
Anonymous attendee asks, short read eliminator was mentioned. Can you compare to blue pippin size selection?
We used to do everything for PacBio with blue pippin. Sometimes you lose all of your DNA. It happens in the gel sometimes. They don't know why. But we've completely shied away from blue pippin, and I exclusively use the short read eliminator for everything I do for long read sequencing now. And one great thing about the short read eliminator kit is that at least I found, if you decide that your yield isn't high enough and you just don't have enough yield to transition to the next step, you can keep your supernatant and pull it back together and do a 1x ampere cleanup, and you can use that as a higher mass. You won't have it won't be size selected, of course, but at least you'll have that higher mass that you can take into the next step if you have a really precious sample.
I actually have never used the short read eliminator. So I noticed that there was an extra large version. What's the upper cutoff? So I like to do like CLR libraries are like 40, 45 kb cutoffs on Blue Pippin. Is that something you can do with the short read eliminator?
Yes, so my coworker will have to correct me if I'm wrong, but I believe the cutoff for the SRE XL is 40 kb.
Nice.
So, yes.
I'm gonna have to get me some.
It's much faster than Blue Pippin.
And I'm sure it's gonna have higher yield. Like you can't really get lower yield than a Blue Pippin. So
right.
It's gotta be better than that.
One thing to consider with this is if you're moving towards PacBio Hi-Fi, you might want a sharper cutoff range at a lower level than the short read eliminator might be able to provide. So it all depends on which sequencing platform you're aiming for when picking your size selection.
Oh, perfect.
That's a good point, yes.
Oh, great points. Oh, here is a question from in regards to Circulomics. Do you have a training video available of the nanobind kits?
We get this question a lot, and we do not have it available now, but we are working on it. So I know that would help a lot of people, and I get questions from customers. I often just take a short video on my iPhone and send it to them, but that is forthcoming. So hopefully that will be a lot more, make things a lot clearer.
Oh, wonderful. And then we have another question from Udayan Umapathy. If there's any recommendations for automation when using HMW extraction and prep.
I think, well, maybe Michelle can talk about this, but I think it would be a little tricky to automate the Circulomics because the position of the disc in the tube is something you really have to pay attention to and manipulate it so that you don't, stick your pipette right through it and crush your DNA. So that might be a little hard for a robot to accommodate.
So that's a really good point. So our Kingfisher kit that we're releasing soon is, well, it's specifically designed for the Kingfisher. So that works differently. It's not liquid handling. It actually is magnets that move. So that's how we circumvent that issue in this high throughput machine. So yes, and that will be able to work on cells, blood, and bacteria.
Well, that's great news. We use the Kingfisher for COVID testing. So we've just been introduced to this amazing machine that doesn't use magnetic beads or pipette tips.
Yeah, it has some real advantages. I love it. Okay, how are we on time? Still have a couple minutes. Fun, lost my place in the question list. All right, Vivian O'Donnell, what about swab samples or oral fluid samples?
I've done reproductive swab samples from guinea pig. Excuse me, hedgehog. I think I used the DNA Easy kit, I believe, but that wasn't for high molecular weight. That was for 16S. But the yield was maybe, well, not yield, but the size was maybe 15 to 20 kb. So that might be sufficient for you.
Okay. All right, our next question is from Jeff Waldbieser. What is your experience with nucleated blood?
Well, nucleated blood is one of my favorite samples to work with. You need very low input to have high yield and great fragment length. So depending on exactly which sample it is, you might have to adjust, but we see from 5 to 10 microliters of input per prep, you can get something around 15 micrograms of very high quality DNA out.
I see that too. I've extracted from turtle blood, which is nucleated, and I believe I used a QIAGEN kit for that. This was a couple years ago though. Michelle, correct me if I'm wrong, but you can now do nucleated blood with Circulomics kits, correct?
Yes, yes,
so
yes. And
if I ever have to do it again, I would use that kit.
Yes, you can, if you have the tissue kit or the CEV kit, you have the necessary reagents, yeah.
So I have a little bit of an interesting experience with the nucleated blood. So I recently did fish blood, and I knew it was nucleated. I wasn't quite sure how to decrease the input. So I thought I'll just do the normal input because I don't know how much lower it's going to be. And the DNA that came out was over 5 micrograms per microliter of concentration. It was a solid blob of gel that took at least a week to homogenize. I actually have a picture, if that's something I can share, because I sent these, Michelle, you might remember, I sent them to her going, oh my God, what did I do? And it ended up being great. It sequenced very well. It was just really difficult to handle, but did anyone want to see what 5,000 nanograms per microliter tube of DNA?
I'd love to see.
Yeah.
Okay.
Oh, perfect. And just to
oh, I don't think I can share my, oh, I can. Yes, here we go. Here we go. So that's what it looks like. It's just a solid plug. Like you could move that tube with vigorous force, which you really shouldn't do, but it didn't move at all. So I had to dilute it like to 1,000 factor to even try to cube it. It was crazy. So I agree with Jennifer that you need a very low input of nucleated blood.
beautiful take
So,
home.
this was 100 microliters, and that was way too much.
Oh, that's funny. I see we are slightly over our time. I feel like we could go on for the whole rest of the day on this topic, but I think we're gonna have to close up the session. To those of you who have asked questions that didn't get addressed in this panel, one of us will write back to you to answer your question. And then otherwise you can always feel free to reach out to me or Christa or any of our panelists perhaps, and we'll answer your questions. We'll try to. And thank you
Thank you
for
so
our
much,
panelists.
Thank you.
Thank you.